Welcome to today's webinar. My name is Lea Valla, and I will be your host for today's discussion with Dr. Philipp Hanisch from Cube Biotech. So welcome, Philipp. It's a pleasure to have you today. We are going to discuss membrane protein challenges and how we can address them using the NativeMP platform from Cube Biotech and multiplexing optical methods from NanoTemper. So I'm really happy to have you because you are the expert. You are a biochemist and you are working on membrane proteins since your master's, I think. And at Cube, you started in 2021 as a scientist, but you quickly became the head of the lab, and you're leading right now the service and purification research team. I think that last year only you purified over 75 unique full-length high-value membrane proteins as drug targets. So I think that's quite impressive, and that's why I'm sure we are going to have a very interesting discussion today. I know that your team is also really eager to develop new techniques every year to improve actually purification steps and also membrane protein stability in general, as those membrane proteins are super challenging for the research right now. And I believe this is how NanoTemper comes in as well to help, as we are developing multiplexing optical methods, in short MOM, for biophysical characterization of proteins and among others membrane proteins. And this combination of multiple optics for the measurements of several parameters in a single assay will enable scientists like you to increase their productivity and confidence in the results that they get with an increased throughput, reduced sample volume, and also less sample usage overall. So I think we are going to explore that together today, and we are going to see how the combination of NanoTemper's multiplexing optical methods and the NativeMP platform from Cube helps membrane protein characterization. So to jump right into it, I would like to ask you actually, Philipp, on your expertise. How do you go on stabilization of membrane proteins at Cube?
Yeah, Lea, thank you very much for the nice introduction. The pressure is up, I can see that. But yeah, of course, I'm happy to answer all your questions today and to go through the webinar together with you. Yeah, here at Cube, we are experts in membrane proteins. Like since the very beginning, since this company was founded in 2012, I think, we are mainly working on membrane proteins, and we have gained lots of expertise. Of course, not only myself, but already our founders did the PhDs in membrane proteins, so there's lots of expertise there. And here at Cube, we developed the NativeMP platform, which is especially designed to address all of these challenges when you're working with membrane proteins. Because as you said, it's notoriously hard to overexpress them, to solubilize them, and to purify them in the end, to keep them stable. And how do we do that? We're providing innovative tools for their solubilization, stabilization, and then in the end, of course, also structural analysis. And the neat thing is we can do all this 100% detergent-free. So we are not reliant anymore on any micelles, on any detergents whatsoever. And one of the central goals of this NativeMP platform is to stabilize the membrane protein in a condition which mimics the physiological environment as close as possible. In short, we want to retain as many lipids as anyhow possible. And this is particularly important for long-term studies where you need increased stability. So for drug screening approaches where you want the highest nativity as anyhow possible, or of course for high resolution techniques like cryo-EM, because we know the lipids are key when it comes to membrane proteins. So how do we do that? The NativeMP platform enables the isolation of these membrane proteins directly from the natural lipid environment. And this we do by using amphipathic copolymers. And those amphipathic copolymers, they can wiggle their way into the membrane and directly isolate the membrane protein with the native lipid environment retained. And some of our audience might have heard already of copolymers, like the early adapters with the SMAs or SMALPs. And in the last 10 years, we've come a long, long way. We had a lot of interesting developments in the field. We're working very closely together with the whole copolymer field. And so as of today, we can offer a broad range of copolymer families and of copolymers in general to stabilize basically every membrane protein you could imagine.
And so That's super interesting. And I think the keyword here that you explain is that you really try to retain the native lipid environment. Can you maybe also tell us how this stabilization using this lipid environment can be used for downstream applications?
Oh yeah, of course. Yeah, I mean, that's the main point. The cool thing is, I mean, usually when people hear of nanodisc, they think of MSP nanodisc, which are derived from E. coli. You purify a membrane protein from detergent, and then you try to press it down in there. You can pick the lipids you want, but what we can do is really come directly from the cell, and this is huge advantages, because by retaining this native lipid environment around the hydrophobic domain of our target protein, we can really dramatically minimize protein denaturation, protein aggregation, and we can preserve this functional activity, which we are really looking for when it comes to drug screening, when it, of course, also comes to structure. And this is super critical for downstream applications, not only structural studies. And our key tools here are definitely that we have optimized with the NativeMP platform these copolymers for a gentle extraction of the membrane proteins. We can maintain their activity and the structure. And so a reconstitution of these membrane proteins into this copolymer nanodisc, retaining this native lipid environment. I cannot tell it often enough. And so we can really try to keep the protein happy. We like to keep the protein where it likes to be, which is with the favorite lipids around. And so what is really important to say is that all of the polymers we have on NativeMP, they are tested in real-life scenarios. We have a big database, as you already said in the introduction, 75 membrane proteins only in last year. And we test all of these membrane proteins with all of our polymers. So we can really say those polymers, they work not only as solubilizers, but they also work as stabilizers. And this is an important thing, I would say. And when I might just add this on the side, it's also very important to watch the tag you are using. So this is also critical, but we might come to this a bit later in the event.
Yes. And so in terms of application, so you mentioned that it's important for, I think, structural studies, but you not only do structural studies, I think you can also tackle at Cube Biotech a lot of applications in drug discovery. And thanks to this NativeMP platform as well, we will also dive a little bit in detail later. What I would like to know is I understand how the lipids and the NativeMP are advantageous over detergents, but do you also see when you use them in the lab other advantages, for example, also when it comes to practical work with your workflow and the steps you have in the lab?
Yeah, of course. So when it comes to membrane proteins, we in the end want to, I think the whole community, we want to describe reality by reality, like how it really is, or keeping it as close as possible to nature, but we have to get the membrane protein somehow out of the membrane, because membrane is drastically complex. There are thousands of protein in there. You have lateral movements. It's really complicated. So we want to try to keep things simple, isolate just what we want to look at, but try to keep it as happy as possible. And using NativeMP gives us a lot of advantages. For example, we do not have to work at 4 degrees anymore. We can bump up the temperature to 37 if you wish, or you can purify at room temperature. And this gives us a lot of advantages, especially in ligand drug screening, for example, when you want or when you need to keep the protein at a higher temperature stable for a longer time. So this is a big advantage here. So we can get like high quality protein samples for binding studies and also for structures. I mean, if you're looking for a new drug, usually drug candidates are membrane proteins. So, I mean, do you want to generate a drug against a misfolded membrane protein? I don't think so. So you want to keep it as happy as possible in the correct conformation, correct oligomerization, and this is where we think NativeMP is the right way. Yeah, and this stands also true for assay development, for example.
Yeah, and so I think you mentioned something about structural work as well. You mentioned cryo-EM at some point and crystallography. I would like to dive also a little bit into this topic, as I know that cryo-EM right now is a technique that is becoming the gold standard for structural determination. So first I want to make a statement, because we are talking about membrane proteins, them being drug targets, but in fact, when it comes to structural studies, there are only, I think, even less than 2% of membrane protein that have been structurally characterized. And this is, we are going to talk about it the whole length of this webinar, but this is because of their challenging nature. So can you maybe comment on some of the challenges and help us understand how you can solve those challenges?
Yeah, of course I would love. Yeah, structural determination. And then when we talk about cryo-EM, when we keep this as gold standard for structural determination next to X-ray crystallography, yeah, it's very hard. As you said, there's only 2% has been structurally characterized. Like often those are mutated. Often you have stabilizing mutants introduced. You have domains cut off, which are aggregating. You have this big issue of even overexpressing them, then getting them out of the membrane. And then let's be real, like no matter how pure or stable or beautiful you think your sample is, which you produced in the wet lab, and I know this very well because I'm there every day, like let's be real, heterogeneity can derail completely your efforts. Like in membrane protein research, especially, like achieving a structural resolution is not about just having a good sample, it's about having the perfectly prepared one. And this stands true from the very beginning to the very end. You can already derail it when you prepare your grid and you have too thick eyes or preferential orientation and all of this. And this is like a real challenge. And I think that's the main reason why, even though 60% of drug targets are membrane proteins, so like we really want it, or industry and everybody, they really want the structures and they want to have the full length, but it's just very hard to get them. So yeah, and like even here, remember, when you extract the membrane protein from its native lipid environment, this is really, you can describe it as a delicate dance if you want. These detergents usually, they strip away the native lipid environment. You can sometimes retain some core lipids, which are very closely interacting with hydrophobic domains, but like the majority is just washed away. And then it leaves you with a mix of membrane protein complexes, fragmented pieces. You have detergent micelles all over the place, and you have to find a way around all this to get rid of the micelles, to get rid of the aggregates, to get rid of the partials of your membrane protein. So yeah, this is a lot of these issues.
A lot of challenges. So now going back to what you mentioned earlier, I think for that at Cube you have a solution in the form of the NativeMP platform, because you are not working with detergent in this case, but with the copolymers. But here I have the next question then for you, because I am not an expert in membrane proteins. So do you have like a way to find the right copolymer? Is there actually, for membrane protein structural studies, one copolymer from your NativeMP platform that works particularly well and particularly fits for such studies? Can you also help us here?
Yes, of course, at least I can try. Yeah. Yeah, here at Cube, even though we are experts in membrane protein purification and stabilization, and we have all this knowledge from like more than 10 years, we also still face a lot of challenges. And like this is why we say screening is like key. Everybody is used to it from detergent times. Everybody was using like 100+ different detergents, screening them all over the place. So we have tried to put like what we think are the best possible copolymers. Those are our own inventions, but there's also the best, what we think from the market is in our NativeMP platform. And we have created a kit, a NativeMP screening kit, all pre-buffered copolymers. You just have to add water or your buffer of choice. And then you can directly start putting this on your membranes, or even putting it on whole cells if you wish so. And then you can stabilize and solubilize in one step your membrane protein. And yeah, we have 5 different backbones, including our own innovation called Cubipol. I think it fits from the name. And this allows to get you really a comprehensive screening to identify the best stabilizing copolymer for your very specific target. And I'm telling you right away, it's not always the same. It varies so much. Screening
So
is
yeah,
key. It's really important.
you were predicting my next question. Like, is there the one? But okay, we know it for sure in science. It's not one size fits all. So actually, I want to come back to the screening process that you are describing using the different 30 different copolymers. What is the next step? Like, how do you then proceed with the screening? What are the different steps that you are taking before achieving a successful structure, for example, with cryo-EM?
Yeah, that's a very good question. So when we look back into old days with detergents, what scientists were doing classically was they were screening the detergents and they were looking for solubilization efficiency. So take your membrane, aliquot them, add the detergents on them, and then spin it down again to pellet all the unsolubilized membrane. And then you just compare, what did I put in and what did I get out? And if it compares, you say like, perfect, 100% solubilization, and this is the detergent I go with. But what we have seen here, and we have seen it over and over times again, not only us, also the whole community sees this, for copolymers, this stands not true. It is not showing you reality, what you see in solubilization. And this is why we go a step further, and we really do a mini screening. Like we take small amounts of membrane pellets or small amounts of cell culture with overexpressed target protein, and then we go the whole route. We purify everything, and for this we typically use antibody resins, and we have our own tech. It's not our own tech, it has been developed in Canada, I think in the 80s. It's the Row 1D4 tech from rhodopsin, and it works like a charm. Like really, it's beautiful. You get super pure results, and with this then you can pretty easily determine what is the perfect copolymer. So how do we do this? Yeah, we purify, and then we of course check on the SDS page, on a Western blot for purity of our protein. Is it not degraded? Is it the right size? But we also tap into powerful capabilities of the Prometheus Panta.
So now that you mentioned the Prometheus Panta is a nanotemper instrument for stability characterization. And so you tap into the capabilities. Can you maybe give us a little bit more detail about how you use the Prometheus Panta exactly?
Yes, of course. Yeah, if you look on the slide, you already see that here screening is indeed key. And even though we have beautiful and really nice clear bands, we still don't know how is the particle which we get out. Because if you look for cryo, like particle size and stability of the complex, of the copolymer membrane protein complex is really tremendously important. So we use the Panta to really quickly identify the best copolymer membrane protein combination for cryo-EM. And how do we do that? Firstly, we use DLS, so dynamic light scattering, to first check and evaluate size and the polydispersity of each complex. Is it in the range we are expecting? Is it way too large? Do we have aggregates in there? Does the PDI fit? And we really get nice PDIs with NativeMP. So often we are in the range of 0.1 to 0.3 in PDI, which is elevated, so it's considered already a monodisperse. And this is great just from elution. But this is not enough. So next we assess how well each copolymer stabilizes the membrane protein. And this we do by measuring the TM, so the melting temperature, with nano-DSF. And with the Panta, we can simultaneously monitor the colloidal stability through DLS and also check for turbidity measurements. And so we can do this over a temperature ramp to quickly assess what is the melting temperature of our membrane protein. And by this, it helps us to rank all of the different combinations to see, do we see any signs of aggregation, and to then like say, okay, this looks great on the gel, it is positive in blot, it looks great in size, it's low in polydispersity, and it's very stable. And then this is the one we go with.
Okay, so now that you have identified the one copolymer that fits this particular membrane protein, what is then the next step before the cryo-EM grid?
So what some scientists are doing, and what you can do, is you can directly take the elution and go in negative stain. And there you can directly see, okay, does it look good? But what we usually do, we are so sure that it's going to look good that we directly upscale everything. So we purify the membrane protein, we run it over a size exclusion chromatography, and then we check different fractions of our sec peak, and then go back to Prometheus Panta and verify all of these key parameters I was referring before. So is the size just right? Does the PEI fit? And so these checks, they really ensure that we're on track for optimal grids. So this really streamlined our workflow because microscopy time is expensive. Like the scientist is expensive. Our cryo expert is particularly expensive. So all of this saves us money, it saves us time, and it also gives us kind of peace of mind.
Yeah, exactly. I think that's a very important point, Philipp, that you're making, is confidence that indeed when you move to the next step, which is cryo-EM, which is expensive or even just time consuming, then you have the confidence that it will indeed work. So just doing a quick summary here, you use the Prometheus Panta DLS nano DSF technologies to screen for the optimal copolymer, and also after you screen to again double check that everything looks good. What are, I mean, you said one of the advantages is that it combines all the different technologies together, but are there some other advantages that you've seen specifically for membrane protein or specifically for your lab as well?
Yeah, it's an easy question. Like we have this device now since 13 months. I think nearly today or tomorrow is the date where it's 13 months in our lab. And I love this device. It was very easy to get to know it, and it requires only minimal sample volume. So what we usually do is we elute in 50 microliter, and a capillary only needs 10 microliter. So you're left with 40 microliter to do your SDS plate, to do your Western blot, to maybe make a mini scale sec if you want that, or to already prep some negative stain. You can everything do out of this very small sample amount. You can work across the broad range of protein concentrations. This is great, so I don't have to like check somehow protein concentration, which can be very hard when you have some peptide in there, or if you have imidazole in there, it's gonna make it really difficult to assess the correct protein concentration. So this is no worry for me. I have no restrictions on buffers. I can just throw whatever I want in there, and it gives me very fast, very high quality data, which I can rely on. At least this is my experience from this. And so all these key parameters I'm really looking for, they are measured simultaneously. So it's a very short time frame. If I'm not doing a temperature ramp, it's basically done 10 minutes, 48 samples in 10 minutes. This is great. So I already know, okay, this is good, this is not good. And then I can make a temperature curve. This takes a little bit longer naturally, but also you're through with this in 45 minutes. So this sets the stage for making informed decisions when I plan my upscale experiments. And also a little anecdote, which I think maybe we even found out in the lab here, which is really cool. If then you do your upscale, or if you want to compare different copolymers in purification efficiency, you can use the initial capillary scan, because the Prometheus, it scans all of the capillaries and it auto detects them. And then it gives you a response unit by a tryptophan fluorescence. And we found out that you can correlate the response units with the actual amount of protein you have in there. Of course, this only works if your protein carries tryptophans, and it works less good when it has very little, and it works better when it has a lot of tryptophans. But with this and your extinction coefficient, you can pretty closely get to the protein concentration. So this is great, and I don't have to do my BSA assay anymore or use the nano drop with every sample one after another. I can just like throw all of them in there, and I directly know it. We've built an Excel list with this. We can just copy it in there. It's beautiful.
So you're really taking full advantage of the Prometheus Panta and getting all these different parameters from it simultaneously, and also with this assay that you now developed for the concentration as well. So I think this is really very nice to hear, first of all, that it helps you optimize, I think, your assay. I also like that you mentioned that with this 10 microliter, then you can do also with the rest of the samples so much other different assay, not just on the Prometheus Panta. So I think that's something also very important to enable scientists to do more with their sample. That's also a super important criteria for us when we develop those multiplexing optical methods. That really all the things you describe is really the way we want to design our instruments. So again, it's really, really nice to hear. And so now I would like to continue explore the different challenges of membrane protein, because we are not through with them, although now we can tackle a lot of them for structural studies with Prometheus Panta. Structural studies are not the only thing that you need to figure out for full characterization. And I know that Cube Biotech really offers a lot of different services, and I would call it almost like 360 characterization. So can you just come back to that and the different application, and almost like if you have some kind of ideal workflow that you can perform with the NativeMP platform?
Yeah, sure. Yeah, as you said correctly, here at Cube, we not only sell products, but we also provide a service, and we do everything from gene to structure and beyond. So this is the nice thing, that we have everything in-house. So the products we develop, we also use them in the lab. So this is why we can quite quickly detect if they're really good or if they're not good, and if they can be introduced into the market. And since our NativeMP platform is a super robust system, and it virtually works or is compatible with all downstream applications, at least I have not found any where it's not compatible with so far, it eliminates a lot of these limitations you typically encounter when you work with detergent-stabilized membrane proteins. And so it opens up possibilities we didn't have before. And I can also give you a few examples. For example, what we've done in collaboration with Proteogenics, we've purified and established a workflow to generate specific antibodies for GPCRs. This is something which was hardly possible, because first of all, in detergent, you cannot or have not seen that you can purify a full-length GPCR unmodified, just like this. It was just not possible. With NativeMP, zack, was possible. We have GLP-1, full-length, works. Then in detergent, if you have to somehow dry your protein or coat it on a plate, in detergent, forget about it. It was not possible. With NativeMP, this works. So we could generate these specific antibodies against P2X4 or different GPCRs, so this is all possible. And this is ideal to produce high-affinity antibodies against native-like membrane proteins, and hopefully also improves the quality of antibodies in future. Yes. Then I already cut a bit to it, immobilization applications. So you can use it for ELISA, you can use it for phage display, binding studies, high throughput screenings. This all becomes now possible. You can do ligand screenings, like, of course, SPR. This works, we have tried it. Spectral shift, also with a nanotemper device, works wonderfully. GCI, we have tested it, works. DEL screening also works. So you see there's a lot of possibilities next to cryo-EM or to structure. Proteomics or mass spec. We have done a big collaboration with the University of Cologne, works also. And then in the end, drug discovery. So this is where all comes together, where you need the structure, where you want to get these small molecule ligands, where you want to create new small molecule ligands. With ASMS, for example, also we did the collaboration, also works out wonderfully. It sounds like a super marketing thing, but we saw stuff which we thought is not possible, but it really becomes possible. Not from today to tomorrow, so you need to invest some time and you need to invest effort, but yes, I'm completely convinced, as you can see. Yeah.
And like you say, it's quite exciting because now it opens a lot of doors in terms of membrane protein research and what's possible that was not previously possible. I want to actually now turn to our audience, to people that are connected today. I see already some questions are coming to us. So before we address the audience's question, I have a question for them, for you that you are watching us. I am curious what you are interested in performing, what kind of characterization you are currently involved on. now, Philipp, you opened the door to many different things people can do thanks to the NativeMP platform, but maybe some of you already are performing some characterization or are interested in the future, maybe with NativeMP. So please let us know what is your interest. And I think this will also help Philipp and I, as we are planning more episodes to this webinar series, to understand you a little bit better and your interest and what we can address to just make the next episodes also relevant, engaging, and interactive. So yeah, I'm gonna give people a bit more time to answer. And maybe, Philipp, I have a question for you, because you are mentioning also throughout this webinar the importance of lipids and how membrane proteins are actually very specific to their environment. Is there maybe some correlation between the copolymer that is optimal for a specific membrane protein and also their environment and their function in cells? And for example, if they are located in a specific area of a cell, if they are, for example, involved in specific function of the body?
Yeah, that's a great question. Yeah, of course, membrane proteins, they can be localized wherever there's a membrane. So it can be core membrane, ER, Golgi, plasma membrane. The nice thing is these copolymers, they dissolve every membrane. So wherever it's going to be localized within the cell, you can extract it. If it's in the ER and you really just want to extract from ER membranes, you could prep your ER membrane before, throw your copolymer on there, and then you're gonna just dissolve this. But you can also directly use the whole cell, dissolve with the copolymer your whole cell, and then extract from all membranes in there.
Thanks, Philipp. So I think now we have the results of the poll. So I'm very curious to see.
Me too.
People are still answering, so I'm not so sure. But for the sake of time and continuing this webinar, and of course having also some time to address people's question, I will continue now and see some of the results. And of course, if you did not answer, but you want to share with us which topics are most interesting for you, you can also use the chat later. So as we can see, we have a lot of different applications as it's expected, but out of the different proposals, we had structural determination, stability assessment, and binding affinity measurements. Most people, 80%, are interested in binding affinity measurements, which I'm very happy, because that's the next topic we are going to touch on today. And of course, because like you said, it's coming all together, right? First, you have to stabilize the protein, then you have to maybe do some structural studies. And what comes next, especially when we are thinking about membrane protein as drug targets, you have to also find ligands or evaluate binding affinities, for example, also you mentioned with antibodies. So I want to talk about that, and I want to go back. Now, you mentioned a lot of the challenges that membrane protein are facing, or that you are facing actually when working with them. So maintaining a native-like environment is key. Of course, we have stability and solubility issues. We also have expression yields. You were talking briefly about that, but can you also go now about challenges that are specific to binding affinity assays? And this could be about the methods that are available to measure the assay, to measure the affinity, but also just, again, inherent to the membrane protein nature itself.
Yeah, of course. Yeah, there's a lot of challenges. You cut through, but yeah, there's more, and I'm happy to answer this. So if we're talking about purifying and having issues of stabilizing it, you're also going to end up with a limited availability of functional membrane proteins. And even if you have successfully expressed and purified them, you have to keep them in a functional state and to get like really accurate binding studies, because how would you know? You know what you put in, but if you don't know if it's 100% active or if it's only 10% active, you're going to end up with different KDs, and this can be a real issue. So make sure that you know your protein is not partially denatured, or it's inactive, else you're going to have unreliable results. then in a lot of binding studies, detergent can interfere in the assay. I mean, the majority still uses detergents. I know that. It has been 25, 30 years detergents being used heavily, and I'm pretty sure they're going to be still used for some time until people figure out that copolymers are maybe the future. But yeah, they can interfere with binding assays. They can mask interaction sites. They can disrupt protein ligand affinities, or they can affect assay readouts. And yeah, this can happen in spectral shift. It can happen in SPR. Then you have, yeah, this comes even on top. Like you have a lack of suitable screening technologies. Like many of these traditional screening methods, they have been developed to detect protein ligand interaction, but for soluble proteins. Yes. And they not fully account for like the unique properties membrane proteins have. Like you have these specialized technologies, like our NativeMP. We really need to stabilize the membrane protein properly, so we can also, in a way, use it as it would be a soluble protein to like wiggle around of this. Else, we will always run issues. Then, of course, reproducibility, false positives. This is true for soluble. This is true for membrane proteins. Yes. It's true for all binding assays. Yeah, and then on top comes all of this complexity of lipid-protein interactions. Like I say it all the time, lipids are important, and they are there for a reason. We know that proteins interact with lipids, or that you have interaction partners which can interact with a membrane protein and with a lipid, or with lipids. So this all, it plays a huge role. And this is why we really need to hold on the native lipid environment as good as we anyhow can.
Yes, that's actually exactly the way you are describing it is holding to the native environment, holding to this lipid environment, but how do you actually do that? Like when you describe, or can you describe like how you go about the binding assay? How do you set up the proper controls and things like that?
Yeah. I mean, of course, when we use NativeMP, when we use copolymers, we don't have any detergents in there, so a lot of these issues already fall away. So we can make sure our protein is stable. We see in cryo that it works also very well, that the structure is still there. We have seen a lot of examples in literature, in the community, that we can retain the natural oligomerization state. Oh my God, I'm missing my words. In copolymers. So in detergents, you often end up with monomers where you should have a dimer, or with like homodimers where you're actually looking for heterodimers. And when you take it out of the membrane, you can keep the lipids. You can also keep the functional state. At least this is true for most or a lot of targets. Of course, there's always going to be examples where this is not the case, but I'm just talking in general. So this already, we can get rid of a lot of these problems. And when we're talking about negative control, for us it's very easy. We can just produce empty nanodiscs with the correct lipid mixture in there, very easily to do that. And then you can use this as a negative control. Then you are sure your target is not binding to your copolymer, or it's not binding to your lipids. And then you can make sure that you can reduce background and get a proper result.
Studying the right interaction. Indeed, yes, yeah. That's super important. And so you mentioned that you are using different methods for your binding assays. So you mentioned, of course, SPR, but also spectral shift, which is one method I would like to discuss a little bit today, because spectral shift is one of the methods that is included in the Monolith X instrument, one of the other multiplexing optical methods that we are providing at Nanotemper. So how do you use the instrument for binding affinity measurements, and anything you can share about the different steps there?
Of course, yeah. We also have the Monolith X here in the lab. We brought it together with the Prometheus Panta. And we have done extensive testing on this, very close to our SPR, which is just standing right next to it, so we can compare a lot. And I really like the Monolith X, I can say this, because you need low concentrations, you need low sample volumes, you have quite quick turnover times. So when you're using chips and you have to immobilize, this is always kind of slow, except you have a device with a lot of channels. So you can use this quick turnover times. It's quite straightforward with the optimization because it's a soluble system, and a lot of these issues with like binding protein to a chip, you don't have this. You can work in solution. You have to label one of the partners, either your target or your ligand, with a red dye. We have a very well-established protocol. We basically just used NanoTemper's protocol and tweaked it a little bit, so it works very nice in our hands. So this could be quickly adapted. We don't have any size or ratio limitations if we're working with big oligomers and very small ligands, which is usually the case when you work with membrane proteins. They are often big oligomers, the trimers, or even more. They're like half a megadalton, or they're like 200 kilodalton, and then you come with a very small ligand of like 200 daltons. And if you don't have the best optimized system and you have not the best SPR in your hands, or you're not the most experienced in this, then you're quickly gonna run into issues, just because the response units are very limited in this area, and then you're gonna have a hard time. And what we see is that especially for these big differences in sizes, the monolith X, it works very well. Sometimes it's a bit hard to convince people that it's also valid, but we'd hope to work on this.
Yes, let's convince them. And so I think what you mentioned also is that this type of big oligomers, small molecule binding affinity measurement is one thing that you can tackle very well with the monolith. But can you mention, or can you share, if you can, other type of interactions that you can measure with membrane proteins? Is there other examples that you've tried?
Yeah, we've really tried it extensively, and we tried to see what's possible. So yeah, of course, the big challenge was these big oligomers, the small ligands. We already discussed this; it works. But if you look into GLP-1R, what is the classical target? Extendin. It's a peptide. We tried this; it also worked. So this was very good. And then we tried, let's try something nobody has tried before. Like, we have the expertise to show a bit off and to produce membrane proteins in their full length. And a nice target is the hepatitis virus. There's like the viral protein, it's a membrane protein. We produce this full length. There's the human receptor, it's also a membrane protein. We also produce this in full length, is NTCP and LHB-SAG, very complicated
I
name.
won't remember the names, but I trust you.
That's fine. So we produce them both in full length, and we put them together, and we could also see an interaction there. Yes. So this was a great success. So we can do basically whatever you want, from big to small, from small to big. All this works.
So that's also very nice to hear such a broad range, and so that really adds also to your capabilities in terms of binding assay measurements, if I understand correctly. Mm-hmm. So I see that we are already at 45 minutes. We will now maybe just also give some time to answer questions from the audience, because we got a lot of them. Before going to the question, I already want to thank you, Philipp, because you have given us a lot of insights. And I want to tell people that this was, of course, the first episode, and we touched a lot of different topics. Briefly, it was a discussion between us, but we are going to dive deeper in all those topics in the next episode. So the next one is going to be more specifically about how to perform the copolymer screening for cryo-EM that Philipp mentioned. And a third episode will be about measuring binding affinities with membrane proteins. So don't or let's say stay tuned and don't hesitate to register for the next episodes if you are curious and want to learn more. So now I'm gonna just go through the questions with Philipp. We have a lot of them, I think at this point more than 20. So I want to also just share a quick disclaimer that we probably won't have the time to answer all of them live today, but Philipp and I are going to gather all the questions, and no matter if we address them live, we will, of course, prepare some answers for you in a written form. Philipp will go through all of them in details, and of course, we will reach out to you from Cube Biotech and or from NanoTemper, and we will make sure that no question is left unanswered, so be reassured. And of course, thank you, the audience, for sharing all those questions. It's super exciting, and I think it also shows the engagement and the excitement. I'm also super excited for the discussion with Philipp, and we are happy that you in the audience seem to be also super interested as well. So I'm gonna start with a first question, and it's about the NativeMP platform, of course. So someone is interested, you mentioned already a few characteristics, but what type of copolymers are included in the NativeMP kit?
Mm-hmm. So in the NativeMP kit, we have 5 different backbones. We call them copolymer families. This is the classical SMA everybody has basically heard about. Is DIPMA. This has been developed more or less in the same time. We have the ASTIs. It's a backbone developed by Anton and Henriette Autzen. We have the ultrasolid amphipoles. They are also known as cyclopoles, and they come from Paris, from the Manuela Zonen's lab. And then as 5th, we have our own copolymer backbone, the Cubipol, which has been developed by our chemists here in-house. And next to these 5 backbones, we have a lot of side chains, how we can adapt this copolymer to generate different physical, chemical properties. So for example, the early adapters SMA, 200 or 300, or basically all of them, they didn't really like to have a lot of divalent cations around them. So like 3 to 4 millimolar was already too much, and then they were aggregating and not working anymore. But a lot of membrane proteins, they require like divalent cations. So for example, our Cubipol or sulfo SMA or sulfo DIPMA, you can throw divalent cations on them as much as you want, and they were not going to do anything. So this is a solution for this. We have copolymers which are stronger charged. They can solubilize in generally a little bit better. We have copolymers which are overall uncharged. This can be interesting if charge is an issue. So the ring itself is then uncharged. And so with this, we really build a kit. If you don't want to think much and you just want to try it out, use the full kit. And if you really have something in your mind, okay, I want this, this, and this property, we also have an option on our website to click on it and to decide, and then you're going to get all the polymers which work with what you have in your mind. So this is like
a tailor-made solution somehow.
Yeah.
Okay, sounds good. So I'm going to move to the next question, Philipp, and it's about... actually, it's a little bit of a challenge for you. So it says, are there any proteins where you have failed to solubilize them in functional form? And someone is looking for tips and tricks to enable, for example, mutation in the proteins.
Mutation in the proteins. Of course. Yeah, of course, there have been proteins we have failed. This is not a 100% success story. As I said, we have achieved a lot in the last years, but there have been a lot of stones we had to climb over.
And of course, that's what's interesting as well.
Yeah, this is also what makes it exciting. Like, what easy world would it be if we just run in here every day and just purify a protein one after another without any issues? Yeah, but what is known in the community and what we also see every time is that if you have a membrane protein which is expressed or localized in cholesterol-enriched microdomains, or you can call them lipid rafts, it's hard to solubilize them. It was already super hard with detergents. It doesn't get so much easier with polymers. Sometimes it can help to increase a bit the temperature, maybe try this, or what can also help is, the key is you need a good expression. If you don't have a good expression in your cells, the best polymer will not do the trick. Yeah. So this is step 1: optimize your expression, and then play a bit around, and maybe try out different cell lines. So this is something which can help. I don't want to go too much into detail, but yeah, of course.
But there are already some nice tips and tricks and some approach to improve the solubilization. So thanks for the insights, Philipp. And of course, feel free to reach out to Philipp and Cube for more insights. I mean, today we have limited time, but I'm sure Philipp is able to share more outside of the webinar.
Really, a little thing I want to add to this. So when it comes to mutations, I am not a big fan of this. We really try to keep it as nature intended it. So mutations, I have a nice example with P2X4, but we can maybe discuss this out of this webinar, which is very insightful, but yeah, try to keep it as natural as possible.
Thanks, Philipp. Thanks for also the precision. So going to the next question, someone I think is also resonating well with the cryo-EM topic. So they are asking if their goal is to go for cryo-EM and not X-ray, what information do they get from the thermal enfolding, because the particle size matters more than thermal stability?
Yeah, of course, you're right. If your only goal is to get the structure, maybe the DTM doesn't count. But if your overall goal is to, on the long run, maybe get also some small molecule ligands, and therefore you have to run it through a specific program where stability is key, then it would make sense to already find something where this also would work with. But if your pure goal is, I want a structure, you're right, particle size, this is what you should look for. Absolutely.
Yeah, and I think one nice thing with the Prometheus Panta, and I want to repeat that because it's something you also actually emphasized, Philipp, is that you can do the DLS measurements at the beginning very quickly, just the DLS measurement for the size analysis, and then you can repeat it along the thermal ramp. So if you do DLS along the thermal ramp, you also get more insight into the colloidal stability, but this is something that runs also simultaneously. So you don't have to do an additional experiment, like it's all at the same time. And if it's only DLS that you're interested in, it's also quite fast. So trying to address as many questions as we are a little bit less than 10 minutes left. So another question, still on the topic of structural characterization, is about differences for membrane proteins purified using the cube, optimal copolymer, or, for example, standard copolymers like SMALP or SMA-LP. I'm not sure.
The SMALP is just the name for the particle. So as soon as you add the SMA and it forms a nanocrystal, then called a SMALP. It's just this lipid particle. That's a very good question. Of course, the classic SMAs are also part of our kit. But yeah, we have seen structural differences. There has been a nice paper of Elisa Moller from NIH, when I recall correctly, where she tried out different copolymers from different copolymer families, and she could see drastic differences in cryo. I think for her specific protein, ultrasolid 18 was not very good. It was exploding the ectodomain, and sulfo SMA worked out nicely. But there are other examples where it can be the other way around. We had very good results with our Cubipol family. And now you say, haha, of course he says that. No, but it's true. We really had good results with this. So they work nicely, but also they are like, it's really the best. Try the whole kit, do a screening once. It takes you a day, it takes you 2 days, and then you know which is the way to go. And I mean, of course you can pick 5, 6 where you think it's the best, but if it doesn't work out, then you have to do it all over again. So
I think, we really also emphasized this, right, when we were saying the idea with this kit is to remove the guesswork, and by doing the screening with Prometheus Panta, you can really just cover the whole range of copolymers available, and you can definitely see the trend in the first step with the particle size and TM, and then once you upscale, you can confirm everything, and once you've done that, you can move forward without a doubt or without second guessing your results. So
what I have always seen since I'm working with membrane proteins, whatever you think you know, you learn something that is not the case. And however you think how smart you are, there's always something you didn't consider. So sometimes it's easier to be a bit naive and just go for it than to overthink everything. But that's just my take.
Thanks, Philipp. No, I like also your approach here. So I want to continue actually on this topic of analytical characterization, because there are some questions related to that as well here. Again, about confirming the results. So how do you make sure the oligomeric state of the protein is correct? Are you using SECMAS? And what are the other functional studies that verify that the protein is active?
Yeah, that's a good question. Yeah, of course, you can use segmals if you are lucky enough to have one. This, of course, implies that you know what is the actual correct oligomeric state. I know there's a few GPCRs where people thought it's a monomer, but then it was a homo or heterodimer. So this also can play our minds a little bit of a trick when we only consider the knowledge we have from detergents or we have from back in the days. And now we see it was maybe not the case. So, but yeah, of course, if you have a segmals and this works very well, you can use this. And of course, if you're in the lucky position to have a very well-studied membrane protein you now want to purify, and you maybe already have a ligand, which you know it has to bind, and there's KD already, then you could check this, monolytics, throw it in there with your protein, confirm. You can do a blue native gel to also see if you have a nice distribution there, but there's several options. Of course, it always considers like you need the protein and you need some time and you need the resources. If you have all this, you can try everything you want.
Thanks, Philipp. So actually moving into the topic of measuring functionality with binding affinity. So there is a question here about that includes SPR. So I'm gonna push it to the slide area for you and for the audience. So they are asking about the nanodisk and if they have a charge, and SPR are typically negative. So how do you navigate static repulsion in this case?
Yeah. I think I also mentioned this already earlier. We do have nanodiscs which overall have a net zero charge, but most of the copolymers are negatively charged. And you need this negative charge to get an efficient solubilization. There are for sure repulsive effects in SPR. What we have developed is also additions to the classical copolymer backbones. We can put a biotin, for example, on the copolymer belt, and we have a few already in our portfolio. And like custom made, we can do every copolymer where it's allowed, so no SMAs. This is just a pattern thing. And this works super nicely in SPR. You can use the biotin and the specific chip to get it properly on there. And we also get quite good adherent rates, I would say. And the nice thing is you don't have to modify your protein itself anyhow. And it can behave as it should there. So this is our take on this. Yeah, but if you're a bit curious or maybe a bit afraid of the charger, if it works, you can use one of our net charge zero polymers.
Thanks, Philipp. Another question, and I think after that I'm gonna address the last question for today. So someone is saying that, sorry, I'm just trying to find the question again. Someone said that they enjoyed the presentation a lot. So thank you, Philipp. I think and thank you for this feedback. The person who asked this question, it's really nice to hear. And they are asking about the purification process. So they are asking if the copolymer is required from the beginning when you purify the membrane proteins, or do you also use the commonly used buffers and then switch to a buffer containing the copolymer?
Okay. Yeah, you only have to use it once. So as I said, no detergent whatsoever, only one buffer. In the very beginning, you use the copolymer. It self-interacts into the membrane. It destabilizes intrinsically the membrane, or we can say it cuts out nanodiscs. And then you just pull on the tag of your specific target protein to a proper purification. You have to not worry about it anymore. You use it as a soluble protein. You don't have to add any more copolymer in there. And so, yeah, this is very easy. Like you cannot forget to put detergent in your buffer, and then there's the level is under CMC, everything aggregates. I mean, I've done this too many times, so this is very helpful.
Yeah, and I think you even have developed a protocol for the purification steps. So this is also something that people can find on the Cube website, and there is even a step on the protocol to, for example, use the Prometheus Panta to also check the stability and that everything is in the right state how you want it. So actually, I see that we are at time, so I want to thank everybody for their questions. Thanks, Philipp, again for all these tips, for explaining the different advantages of the NativeMP platform, for explaining your workflow in details and how to tackle all the challenges of membrane proteins. Thank you for the audience for engaging with us, answering the poll, and we will see you on May 8th for the second episode of this webinar series, and it will be about screening the copolymers for cryo-EM with Prometheus Panta. So yeah, it was a pleasure.
It was a great common effort, and thanks everybody for attending.